In this video, we will discuss about some of the in vivo studies of the bacterial delivery system, that is a cap-based delivery system that we have discussed in our previous videos. So, in order to check the in vivo system, what it is, they intravenously inject their bacteria with this induced cap and also with the knockdown of this KIFC to find out which are the cytokines that are upregulated. And what they are able to see, that in the case where KIFC gene is knocked down, they are able to see more cytokines are formed. For example, this interleukin 1 beta, IL-6, interleukin 10, GCSF, and GMCSF. However, they are usually low in the case of EC and iCAP. Especially in this interleukin 1 beta, it's significantly lower, and also this interleukin 10 and GCSF, which are significantly lower compared to the case where ECN with this KFC gene is knocked down, indicating that the immune response generated by this ECN iCAP is lower compared to the case where the ECN in which the cap is not there. So, and this is in line, these results are in line with the results that they have seen in the in vitro studies. Next, they wanted to check the dose dependent effect of their bacterial delivery system and check its impact on the survival rate and relative body functions, relative body weight. So first, so what it did is they intravenously inject this cap bacteria to check the dose escalation toxicity. First they check the body weight, relative body weight, and they are able to see that in the case of EC and iCAP, the decrease in the body weight is significantly lower compared to the case where wild type ECN is used. And when it comes to survival rate, the induced iCAP ECN has a higher survival rate, and there are no death observed even after 2 days. But for the case where ECN is used, they are able to see a decrease in the survival rate just after 2 days, and also for the ECN with KFC in knockdown, the survival rate is decreased. We can see that after 2 days. And when they check its toxicity fractions, what they are able to see that toxicity fractions are higher for the case where they use wild type ECN or ECN with KFC gene knockdown. But when they use the ECN with iCAP induced, they are able to see a low toxicity compared to the other two cases. Next, they wanted to check the maximum tolerance, and in order to do that, what they did is they take the mice and you inject them with this bacterial delivery system, and also they inject this theta toxin uh, that basically you can say that it it uh, theta toxin actually enhances uh, the replicability or this system. This theta toxin delivery will enhance the bacterial delivery systems, and what they are able to find out in this case is that in terms of the efficiencies, the iCAP systems has more better efficiencies compared to the case where the ECN without iCAP is used, and this is true for both uh, this in terms of the toxicity and also for the efficiencies. And we can see that the response is high at lower dose for the iCAP induced cells, but for the other cells uh, response is slower. And these things they have quantified in this bacterial luminescence while targeting, and they use the theta toxin delivery so that this model, this panel K model, is to target they wanted to check whether their bacterial systems can target at the tumor sites or not. And this is what they are able to find out, that iCAP system's response is better compared to the case where they don't have the iCAP system. And the bacterial luminescence images configurations are shown in the panel L, where we can see that when they use the ECN iCAP, they are able to see a high bacterial luminescence, whereas when they don't use the ECN iCAP, or the low concentration of ECN iCAP is used, they are able to see less bacterial luminescence, and the difference is statistically significant. And in terms of the tumor growth also, they are able to see a 400% decrease in the weight of tumor growth after the 15 days for the case where they use ECN iCAP compared to the control case. And the results for the panel M is shown for the colorectal CD26, and similar kind of results they have seen it for the genetic breast PYMT or MMTV, PYMT MMTV cancer cells. Here also they are able to see 100% decrease in the case of where they use 100% decrease in the tumor growth, 100-time decrease in the tumor growth for the case where ECN iCAP is used compared to the case where ECN wild type is used. So this altogether indicates that their in vivo delivery of this CAP system works well compared to the existing systems that use the wild type ECN, or if there is no ECN treatment is there. That's what they have concluded in this results that are presented in this figure. If you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.